So genic balance system, as we have told that this system was developed by Bridges in 1922 in Drosophila. According to this theory, or we can say that the summary of the genic balance theory of the Bridges is of the following. So according to this theory, the sex of an individual is determined by a balance system. The genes for maleness and those for femaleness present in the individual. So genic balance theory states that sex determining genes are present on both X chromosomes as well as on autosome. Here we can say that the Y6 chromosome does not have any roles for the development of maleness in Drosophila. So the male sex determining genes are present on autosomes and female sex determining genes on a X chromosome. So the sex expression is determined by the balance of genes on autosomes and X chromosome. In other words, the expression of sex depends on the ratio of X chromosome to that of autosomes. This ratio is represented as a X/A ratio. The genic balance is governed by X/A index. Individuals with index of 1 develop into female and those with 6 index of 0.5 into male. And these male and female are normal. If the 6 index is between 1 and 0 and 0.5, the resulting individual will be neither a female nor a male, but have an intermediate sex expression and it is called intersex. The sex index of 1.5, which is higher than the sex index of normal female, gave rise to super female, and a sex index of less than 0.5 is lower than the sex index of normal male gave rise to super male. So here you can see in the table that 1 6 index ratio normal female will be developed, and 0.5 6 index ratio normal male will be developed. In between 0.5 and 1 6 index ratio intersex will be developed, and below 0.5 super male is developed, and above 1 6 index super female is developed. So here this can be easily calculated that 6 index ratio is number of X chromosome divided by number of autosomes. So individual with the 6 index of 1s are normal females, and flies having the 6 index of 0.5s are normal males. And this theory is known as genic balance theory, which is represented by Bridges. And these are the summary of genic balance theory. Monogenic sex determination. So expression of sex which is influenced by a single gene is known as monogenic sex determination. Mono mean one, genic mean genes. So the expression of sex by a single gene is known as monogenic sex determination. For example, in Drosophila, transformer genes which is present on autosome play an important role in sex expression. So transformer gene is recessive and hence does not have any effect in heterozygous condition on either sex, in male and female. In a homozygous condition, this gene transforms the normal diploid female into sterile males. Single gene control of sex. So single genes are found to be responsible for the determination or expression of sex in Chlamydomonas, Neurospora, yeast, asparagus, maize, as well as in Drosophila. Monogenic sex determination in maize is an example. You know that maize is a monoecious plant with male inflorescence, which is known as tassel. In female inflorescence, a silk is located on the same plants, and this plant is known as monoecious. A gene called tassel seeds convert the tassel into the seed-bearing inflorescence. Another gene, silk lesser, is responsible for absence of silk. A plant with genotype SK/SK will be effectively a male plant, and plant with genotype TS/TS will be effectively a female plant. So the individual single genes SK/TS can impose bisexuality in a maize. Monogenic sex determination is in Drosophila, as I have told that transformer genes in above. A similar SRY gene is found in human, and this gene is probably present on autosome and transform the normal male into female. Such male have feminine, female characters. They have internal degenerated testes and are sterile. This condition is known as testicular feminization. Similar effect of single recessive gene on expression of sex have been found in some other animals such as goat, pigs, dogs, etc. Environmental sex determination. As we know that genes always react with the environment to produce the phenotypes. So the environment have their own role for the determination of sex of an individual in some organism. For example, sex determination in some organisms such as sea worms, Bonilia and Dinophilus, and horsetail plant which is known as equisetum is governed by environmental condition and also includes some hormonal effects. In Bonilia, the larvae which remain free in sea water and settle on the sea bottom are differentiated into female, while those larvae which settles on the proboscis, a female, develop into tiny males. Incubation temperature, that we also said that environment have their own role for the determination of the sex of individual. So for example, in turtle, alligators and crocodiles, you know that alligator, crocodiles and turtles, they belong to the reptiles. If the incubation temperature of egg is high, for example 30 degrees centigrade to 35 degrees centigrade, female will be developed. If the egg have temperature, incubation temperature 23 to 28, male will be developed. That's why we can say that environment have their own role in the determination of the sex. Sex mosaic in Drosophila. Combination of male and female features in the body of an individual. Drosophila flies 1 in 2000 have male tissues in one part of the body and female tissue in other. So individuals with such sex mosaics are known as gynandromorph or gynanders. You can see here in the picture that one or half part of the Drosophila is male and half part of the is female. So this type of Drosophila is known as gynandromorphs or gynanders. And the three patterns of sex mosaics may be found in Drosophila. Bilateral sex mosaic. Mosaics mean dagdar, the bedar. So one side of the fly is male and the other side is female. This is the most common type of sex mosaics. Antero-posterior sex mosaic. The front half of the fly is one sex and the rear, or we can say that posterior part of the body is of other sex. Sex mosaics and patches. Sometimes only a quarter of the body is male and rest is a female, or just a small patch of tissue may be male and rest of the body is a female. This process is also known as a sex reversions, and these have these type gynandromorph. A gynandromorph is an organism that contain both male and female characters. The term gynandromorph from Greek word gyne mean female, andro mean male, and morph mean form. Those individuals having both male and female characteristics, they are known as gynandromorph. So depending upon the position of sex tissue, there are following types. Bilateral gynanders. Sometimes one half of the body show female characters while other half show male characters. Interior, posterior, that interior have one sex and posterior have other sex. And six piebald's. So body consists of female tissue having spots of male tissue scattered irregularly. You can see here that this is known as bilateral gynanders. This is known as anterior posterior gynanders, and these are known as six piebald's. Sex determination help in selection of plants for crop improvement. Sex determination help in identification of plants by which it will save the time. For example, in nutmeg and kokum. If you like the video, please share, subscribe and watch. Thank you.